much for registering, showing your enthusiasm in learning. I mean, getting to know more about new drug development opportunities and showing your interest to hear from the experts. That's more important. We are only coordinating. I'm Dr. Pratibha Nadig. I'm founder director of Akshay Sarthi Healthcare Solution. And today is a master class. It's a webinar on new drug discovery and development, which will actually give an overview also, and few aspects will be covered only in this session, which will not be covered later. We have made it open for everyone so that everybody gets an idea. Some of you may be already aware of few things, but maybe we could interact, add more, or learn from each other. That is how it's going to be. And that will be the only session today. Today, it's going to be very interesting because you have a member from your peer group, and he's quite popular, I heard. And my purpose of calling the guest today, Dr. Harshad Ramineni, all of you know, and many of you have joined because he is there. And so why I called him was, one is his MD pharmacology, and he will definitely share few things which you can very well connect with. Second thing is, he is coming from my own institution where I graduated, did post-graduation, and I worked for a few years, and that is an excellent academic institution. He represents one of the premier institutions in Karnataka as well as in the country. Yeah, you can mute yourselves, all of you. So we have Dr. Harshad Ramineni. Can you come on the video, Dr. Harshad? Are you there?
Yes, sir.
Yes. So he is founder of Aura Farm Lab, an academic studio for medical postgraduates. He is creator of Pharma Notes, Pro Exam Shaped MD Pharmacology Notes, 6,000 past questions indexed across 7 universities. He has built PG answer banks across multiple specialties: pharmacology, medicine, OBG, forensic, and dermatology. He is an MD in pharmacology pursuing from KMCRI. When I did, it was KMC Hubli, Karnataka Medical College. Then it became Karnataka Institute of Medical Sciences. Now people of Hubli have forced them to make it Karnataka Medical College and Research Institute, Hubballi, affiliated to RGHS. He is an undergraduate from Seth GS Medical College, Mumbai. So what attracted me to his candidature was, as a student of MD pharmacology, he has been doing something beyond his area. He's trying to help others, trying to uplift. So that very well aligns with what we think also. But Dr. Ramneni is doing everything free of cost, I believe. He is not charging, and we charge. And the purpose of charging is two ways. One is, when you charge, students become serious about it. You become adherent. I can question you, and I feel accountable to you. I'm attached to you. That's what has happened in the previous batch. And second thing is, we are totally independent private organization, but we are trying very hard to give you accreditations with universities, medical council, credit hours. We want to make this course a very robust one and try to get the best faculty so that they interact with you and you do justice to the subject which you have taken. That is what is my concern. Because today is going to be a little relaxed session. When I started my career in Natural Remedies R&D Center, I completed MD in KMC Hubli. Best teachers we had, but limited resources, limited infrastructure. Bioassay with guinea pig ileum we had done. When I came here, they said we have to find out a new herbal combination which acts as antihistaminic, and you have to set up, and you have to have metrics. You should show its potency, you should show its efficacy. I was not competent enough, and I took a lot of time to learn by myself, and the team, which was not only physicians, it was biotechnologists, biochemists, and they were all BSc, MSc, PhDs. The way we worked together, and I learned from them. Great learning it was. So my journey started from there. I will not go into too many details, but nevertheless, I would like to introduce that I worked in R&D center. I learned preclinical pharmacology, then clinical trials, And then I did a PI. As a PI, I have worked as a principal investigator for nutraceutical trial. Then we did lot of protocol development as per GCP guideline. I headed ICRI, Institute of Clinical Research India, with Cranfield University. We brought standards in clinical research. We trained so many MSc clinical research who are all my students are in best position all over the world. Then I came back to Vaidehi. Then we have continued with research coordination, postgraduate teaching, PhD. I had a PhD candidate. Now I have become an independent innovator. So my exposure has been wide, and I thought it's worthwhile to share. I will not stop sharing whatever be the number of entries or registrations. Doesn't stop me there. Our team is very good. I am joined by Dr. Nanjaraje Aras. He's our CEO. He will join in a while. And we have so many, I will explain about that later. Now, with that, we get going with the course, with this thing. Before that, Dr. Ramneni, I have called here because he's a senior person. He has crossed an extra mile, as I told you. So, Dr. Ramneni, just one question before I start. We have so many MD pharmacologists. Is there anybody pharmacologists who are M-Pharma? Anybody in the group? Can you please unmute and answer? Anybody among the registered participant who is not an MD pharmacologist? Because I want representation across disciplines, not only MD students. Anybody is there? Any, I think Dr. Somebody had approached today. That's okay. Anybody is there representing companies? Somebody had inquired today from Vivint. Samir, I think. And Samir, Dr. Samir is there? No, okay, that's fine. So anyway, if that is not there, that should be okay. Dr. Harshad, when people join, so many have joined pharmacology. Are they very clear? What is your understanding about the people who join MD pharmacology? Are they aware of what they are going to do after their completion, where they are going to be? With that complete preparation, do they join MD pharmacology, or are there any other reasons?
My own experience is I had joined because I knew that I wanted to get into corporate world. So pharma companies, I knew that. But otherwise, I think most people, I think more than 50%, would not be knowing or uh, would not have a preference for clinical branches. For that reason, they chose non-clinical branch like pharmacology. And some may not even know uh, what what the curriculum of pharmacology is, what the perspectives are in the future like. So they
Okay.
just join, but a small part they are very clear that this is what they want to do.
This is what they want. How did you get exposed to corporate world when you were an undergraduate in State
Uh,
Medical College? How was it?
yeah. We were involved in the cultural events, so those were sponsored by the pharma company. So we had interactions with the
Okay.
pharma
Okay.
companies. Yeah.
So getting exposed as an undergraduate to all the opportunities would be worthwhile, I
Yeah.
think so. So that they can choose,
Yeah.
because pharma cannot be undermined. It is as good as clinical
Yeah.
subject. Do you agree? Yeah,
Yes.
you will definitely appreciate that as we run through, and you will also have an experience. Anybody else
Mhm.
want to add on to this? We'll come to other discussions with him as we run through this, our presentation. Anybody else has anything to say regarding this? Were you aware? How many of you are aware? If you're very clear and then joined with the right aptitude and you wanted to be in pharmac, anybody in the group, you can be free and you should be free. You should be open. Let's have interactive, because as it is online is not very, it becomes a little monotonous if you don't interact, right? Anybody wants to answer? Yes, please. Vinod.
Fine.
Yes, Vinod. Vinod is a Amit. Amit wants to answer. Vinod or Amit, who
Yes,
wants to
ma'am.
answer?
Good evening,
Yes,
ma'am.
Vinod.
Good
You
evening,
can go
everyone,
ahead.
ma'am.
Good evening. Good evening.
Yeah, yes, like even I was actually very clear that okay, I have to go to the pharmacology. As like Harshad sir, he also told me the same thing. Like even I was also like very fascinating about the corporate life, everything. And what I thought was, ma'am, like when we became a physician, so I could able to treat only a set of patients or something like that. So when if I become a pharma, or like if I go into drug development or in a industrial level, so I could have, I thought like, okay, I can make some big difference. And
Okay.
the life was also non-hectic, and it was no emergencies. That was also like
Okay.
most
That was
one of
good.
the major concern also, ma'am. So
Okay.
therefore I chose pharma.
Okay.
And we can also do so many other things other than pharma, other than clinics. Like if we are in
Okay.
pharma, so like not only the like the patient and the disease everything, so we can go, we can like we can spread or like spread everything, like so that example like Harshad sir, so he has done so many of
Hmm.
startups, entrepreneurship, everything. So I thought like, yeah, so even I thought like pharmacology also, it's very helpful and it is very easy for us, and we just
Yes.
joined.
Great, great, great. Very good. Yeah. And therefore, you and Harshad both are already entrepreneurs. We can discuss that
Thank you.
again when we come to the last session.
Sure, ma'am.
Great going. Yes.
Thank
Jawahar,
you.
is it Dr. Jawahar? Thank you, Vinod. Dr. Jawahar, anything you want to say? You have raised your hand. Amit Bhatt,
Yes, good evening, ma'am. I'm Dr.
good
Amit.
evening.
I'm a
Yes.
second year MD student from Father Medical College, Bangalore.
Bangalore. Very
And
good.
yeah, I'm based in Bangalore. And yeah, I've taken pharmacology because I've loved the subject since the time I was a second year. The magic
Okay.
of how a single pill, when it is prescribed, the patient automatically feels better after the course for which it's given.
Okay.
It's just, amusing to watch.
Very great.
And so I wanted to know more about exactly how the process happens, how
Yes.
PKPD happens, and the downside was when we take a branch like this, uh, the people we approach for guidance, they're very
Yes.
limited. They just say, okay, you have research, you have teaching, can join a company, or there is a fourth option. But I mean, they list it out, but there is no insight on exactly what happens when
Yes.
each of these avenues are spoken about.
Correct.
So the goal is to explore
Right.
exactly what comes under each of these four avenues. And it's a pleasure being a part of this meeting. Thank you for organizing this.
Great, great, Jawahar. Very nice. So you also had some exposure and you came with, will to do particularly pharmacology. Not that you did not get clinical, so you are here. It's not that way.
Absolutely not. No, ma'am. I
It's not
wanted
that way.
pharma and I was fortunate to get it.
Very good, very good. Yes, doctor. Thank you so much, Dr. Jawahar. Are you there, Dr. Jawahar? Would you like to express something? If not, we get going. Okay. So with this preliminary thing, so I understand some of you many of you would have come with interest, but you are not expressing. As we go through the sessions, you can open up. Don't hide, and let there not be any hesitation in expressing. And let's be open. And I would love interaction. Without interaction, it becomes very monotonous. And okay, my slides are visible, right? Okay.
Shiva madam, can I add?
Nanjaraj, yes, yes,
Yeah,
most welcome. Before that, I will introduce Dr. Nanjaraj is our CEO, and welcome, Nanjaraj. Over to you. You can
yeah.
express.
Thank you, madam. Good evening, everyone. Just I wanted to, share, my own perspective. I started my career as a wet lab in a, herbal pharmaceutical company. Then jumped into, biotech pharma. From there, went on search of my career, the curve in the contract research organization in the Indian company and in a MNC company, and finally ended up into the general management of the R&D projects and other things for a big pharma. Here I wanted to add, been really blessed to have a mentors like Pratibha madam. They guided me. Even though I feel I'm being fast tracked, the efforts I put in or the knowledge which I've gained over a period of years could have been much better if I had a orientation in the beginning itself, what this drug discovery is all about and where all it can be applied, because it's a super specialized area. It can be applied in the pharmaceuticals, it can be applied in the lab, It can be applied in the contract research organizations. It's a vast area itself. So we could have put in a little more focused effort to reach our goal. So this course is being curated so fantastically that you will get a learnings via online classroom, and you will also get to visit all the probably 3 or 4 facilities, like the what is happening in the R&D, what's happening in the BAB center, what's happening in the manufacturing part. So that will give you a glimpse where you personally feel you exactly fit into that role, or you are passionate about exploring more. So that's the beauty of it. And thanks, Pratibha Madam, for giving me opportunity to speak here. And
Yeah.
wish you all the best to everyone.
Thanks, Nanjaraj. And Nanjaraj has had a very wonderful journey. We were together in the R&D. From there, I moved to academics and other side, and then he moved on to CROs, Clean with Clean no, Clean Track,
Clean Invent,
Clean Invent
and
with
then
Arun
let
Bhatt.
me,
Yeah, yeah. Pioneer, Dr. Arun Bhatt from Mumbai. He's a pioneer in CRO. Then he led the PPD, IQVIA, managing the entire Asia Pacific region. He has got a very rich experience. He will be speaking in one of the sessions. And my association with him, I have also learned so many things, excellent management, including financial operations, everything, he has had. So I'm also lucky to have him by my side. We have been together, though in different organizations for the past 20-25 years, and we have joined again as together. We are co-working, right? So with that brief introduction, let's start today's session. So today's topic is to give overview and discuss few aspects in depth about new drug discovery and development. We may think that new drug discovery is only innovation and limited to the lab scale. No, we are going to discuss from the time it is conceived by the discoverer, that is a sponsor or pharmaceutical manufacturer, company owner like Cipla or Roche, all these companies, So Biocon. So these are the ones who are innovators. So from the time it starts there and throughout its life cycle, that means it remains, it gets developed in the lab, in the animals, in the clinics and clinical trials. It reaches the clinics, it remains there under observations as long as it is there with the company. And after the patent expires, somebody else will also start manufacturing. But if the drug is a wonderful molecule such as paracetamol, which we have today, it remains there forever, for decades to come. So the entire thing constitutes new drug discovery and development. I would like to emphasize that it refers to the entire process of development and its life cycle until it goes out of the market or it is banned or whatever. The entire period should be taken as drug development. So, we discussed, everybody wanted to know what are the different careers. I will start here and again come back to this slide. Once you complete your MD pharmacology or M Pharma PhD or biotechnology, any of these, there are some specific areas where medical pharmacologists are required. Elsewhere, even M Pharma pharmacology PhDs are also able to compete, or you can say they share these roles, So medical affairs, where you bridge the science, evidence, and healthcare, you represent the company and you will bring it to the table, clinician's table. There you are talking technically, the research, the observations. Will be familiar with the publications, you'll be familiar with the reports, statistical analysis. You will explain to them in a way that it will give them confidence about the evidence this drug has produced through clinical trials, through preclinical, through comparators, and in special conditions. All those you will be explaining. So you need to master the art of communication as well as have a thorough knowledge of interpreting the publications. Interpreting, that is why you have journal club, that is why you have seminars, you have communication skills. Pharmacovigilance will ensure that academic institutions also, they have put you to collect the ADR reports from the hospitals, and processing of these ADR reports, individual case report forms, ICS, individual case study reports, ICSR. So these will be received by the contractual research organizations such as Accenture, IQVIA. All these are into pharmacovigilance. Many more companies are there. Bioclinica is there. So these companies, they get the reports from various sources, and normally these are doing the work for international companies. Those companies will send, whatever they have received by the general public, and that has to be processed, and you will be trained there. You will be asked to do the causality assessment. If you know what it is, that means evaluate the ADR, whether it is truly an ADR or just a false report. Is it definitely related to the drug or due to some other reasons? And all these things, processing means attributing whether it is due to the drug or not, because it's very essential for the pharma manufacturer to know whether his drug is performing properly, or what are the ADR, what is the severity, how many ADRs are there, because the government will keep a watch, to see where if it increases in frequency, then they'll have to, withdraw the drug from the shelf or ban it. So very, very important industry, multi-billion industries, pharmacovigilance. And medical writing, some people are very good in writing publications, reports, protocols, and even the pharmacovigilance part of writing, regulatory submissions. Different types of medical writing is there, which companies will outsource. So they will hire you, and if you have a good writing skill, you will do all those things. Regulatory affairs, very few MD pharmacologists are entering, but one of the wonderful areas, CDSU had also called for some positions in regulatory affairs, where you will be, if you sit on the government side, you will be reviewing that. You'll review the submissions which you get from the pharma industry. If you sit as a part of the pharma industry, you will prepare the documents. It's more of documentation. So pharmacologists are mostly involved, not on the field, but we use our intelligence and brain and our written skills to develop these regulatory submissions, which will adhere to that country requirement. Think globally. You will not be adhering to only Indian regulations. You will have to make the documents which suit international requirements. And being a pharmacologist, you will be able to word it very clearly, because you know what is pharmacodynamics, you know the dose response, you know what is the maximum tolerated dose. We are going to discuss all that, and that's the whole purpose of having this course. So once you know these terminologies and you're familiarized with that, very easily you can write, and of course every company will train you. Then there is R&D, where I have worked. So R&D is behind the screen, less glamorous, but for those who have an aptitude, you can, very, very, very interesting, where new molecules are, we'll see how it progresses. Molecules are designed, then you have to test in cell culture or tissues or lab on chip nowadays. If you go abroad, Stanford, Harvard universities have developed wonderful labs for that. And very interesting to see how to pick up the best molecule there, develop assay systems, enzyme assays, all those, R&D, where you can come out with your drug. After working for 10 years, maybe you can have your own setup and come out with your own molecule. It's very interesting. Otherwise, you will be part of the innovator company. That also is very, very satisfying. Then another thing is academic research. Some of you, many women usually, no gender bias here, and some men also, can prefer being in academics to balance work-life balance, family responsibilities. That's how I also chose for many years. I was in academics only. So, but here also there is a lot of scope, and you must not relax only teaching. Teaching is wonderful, but there in academic research, now the ecosystem in India is so good that you get a lot of funding. You can do your own research along with teaching, and institutions and universities are supporting you. We shall see in detail what these are all. How you can actually contribute in each of these areas. So have a clear idea that these are all the things, depending on what actually you want to do. Frankly speaking, all these are, you should gain hands-on experience to understand the functioning of a company. Once you understand for 5 to 7 years, then you are free to take up whatever you want to do. But first understand and pick up the areas and never find it monotonous. Okay. So with that, now we will start with, I'm going to start with basics because some students have just joined. I will start, it may be repetition, nevertheless, let me also get familiarized once again. What is a drug? Any chemical substance other than a nutrient or essential dietary ingredient other than nutrient, when administered to a living organism, produces a biological effect. When you administer, it should show a response. That's it. WHO says little, it makes it complex. Any substance used to or intended to be used to modify or explore physiological systems or pathological states. In other words, any agent which can explore, that means which can be used as an agent for investigations or to treat pathological states to diseases, that is defined by WHO as a drug. And medications have different dosage forms. Different dosage form and formulation are used interchangeably, but actually formulation means what goes along with the active drug. Active drug with the excipient constitutes the formulation. So this is what we have learned as fundamentals. But nowadays I see people use dosage form. Solid dosage form is also formulation. Liquid is a form. Obviously, the excipients used in solid dosage form, such as the tablet, all different types of tablet we have shown here, chewable, dispersible, vaginal tablet, all these are different dosage forms in solid dosage forms, oral as well as vaginal. Apart from that, we have others. This you have discussed. I need not go through, your teaching students. Then drugs can be given by different delivery systems. You can administer the drug in different novel drug delivery systems. You know about that. It could be a transdermal patch. It could be a pump. It could be a targeted drug delivery. It could be liposomal. So in various ways, the drugs can be given. So much of activity is happening with drug development. Simple dosage forms, there is a lot of research. Oral solid dosage forms, so many big spectrum. Delivery systems, there is a big spectrum. And then we have why this is important, because you should know when you deal with drugs, what is a branded drug company is the sole innovator, patent holder. He does lot of R&D, which goes in several years, and several $billion are invested. Naturally, he will own, like Pfizer, Merck, AstraZeneca, Johnson. They have lot of patented drugs because that gives them a lot of royalty. We can sell it and get huge money. Most of the pharma industry's intention is to bring change in the healthcare, but they are out and out commercial organizations. They need to take care of finances very strongly. So they are branded drugs, where nobody else can copy. Then generic is once they lose the patent, 15 years, 20 years, then anybody out of patent, Indian companies, we have many companies which will manufacture the same drug because they're out of patent. So they manufacture different formulations. They purchase an active drug, active ingredient, and they make their own brands. That is branded generic. But otherwise also generic is Dr. Reddy's, Lupin, Torrent, just generic, unbranded. There is no brand there. It just carries the name of the chemical name of the drug, paracetamol, ibuprofen, low cost. But the quality can vary because they are usually very, very cost effective, economical drugs. Branded generics are generics which are manufactured by Cipla for a Pfizer drug when the patent goes off, but they are very well supervised and they carry their brand name. So these are called branded generics. This is how different drugs are available in the market. Brief overview of kinetics and dynamics. You people have appeared for NEET. You have started reading. I hope you have started reading, and I had put it in the group that you should come prepared. So kinetics is absorption, distribution, metabolism, and excretion. All these processes, absorption takes place in the stomach for oral, the intestines for oral drugs, and at the site of administration for injectable. Then it gets distributed. As it gets distributed after absorption, portal goes to the heart, then it gets distributed across, then reaches its site of action. During that process, it gets metabolized and excreted. So as it gets distributed, it reaches a site of action, produces clinical effects, which is dose dependent. As the dose is increased, the response goes on increasing. As the dose is increased, also the concentration increases. That comes under kinetics. Response comes under dynamics. So here we plot plasma concentration curve with time. Here we plot a dose versus response curve. This we have to be clear because we are going to discuss few aspects in the coming sessions. So I hope so far there is no difficulty in understanding. And we all know that drug works through drug receptor interaction. That receptor which is implicated in a particular disease, for example, sulfonylurea receptor, we want for sulfonylurea drug because we want to enhance insulin secretion. That becomes a target, that is a receptor for sulfonylureas. So every drug has got a target, has got a receptor which is considered to be a target, which is involved in the disease, which is involved, or combination will help the disease. But basically a drug combines with its receptor and it is specific. That is also there. We are going to learn that how they are made very specific, like a lock and key almost, for drug receptor interaction. So we know that drugs work by drug receptor interaction, and when the drug combines with the receptor, lot of intracellular changes take place that results in the ultimate response. And you know, this picture I have taken as it is from Tripathi, that there are different types, receptors which are cell bound. One of the these are few receptors actually, if you take, but majorly when we study pharmacology, we know that drug can combine with receptor which belongs to one of the four families. Either it is an ion channel, or it is enzyme related, an enzyme bound to the membrane, or it could be G protein coupled receptor, or it could be nuclear receptor such as steroids. So this one we normally say steroid receptor, this one insulin receptor. Here we for GABA or benzodiazepines, and G protein for adrenaline and other drugs. So like that we know about the drugs. Now we shall understand how are these receptors characterized and how they are identified in particular disease. Only then we can bring out a new drug. So am I clear? Is my communication okay with all of you? Should I go faster or slower? Can anybody unmute and say?
All good,
Hiranna,
ma'am. Very clear.
good? Okay,
Good.
thank you. Yeah. So now let us understand what is a new drug. According to, I'm now introducing you to the CDSCO. What is CDSCO? Central Drugs Standards Control Organization. This is the highest authority, central licensing authority, who give permissions for the sponsor to market his drug. Government of India, they have defined. If you go through, I will come to that regulatory, document which you should read. They define a new drug as what we normally understand. First time it's introduced, that has not been used or approved as safe and effective by the Central Licensing Authority in India. This is one of the definition of new drug according to regulation. Now we have a drug, but they say I have introduced this drug for diabetes, but this drug also helps in PCOD. Anybody? Which drug is that?
Metformin.
Anybody?
Metformin.
Very good, very good. So like that, if you modify the claim, then don't start marketing it on your own, or let the physician not prescribe it on his own. It becomes a new drug. Again, it has certain requirements. You should show that it is safe and efficacious at a particular dose with minimal toxicity in PCOD. So when you modify the claim means I would have introduced for something, now I have a different claim for a different therapeutic indication. Then any new indication becomes a new drug, although it's already there. So can you tell me what is repurposed drug? Have
A
you
drug
heard of
which is
it?
already used for one purpose, but not
Yes.
identified for any other purpose, but then we are using it for other purpose. It is called repurposed drug.
Yes, yes. So that
Example,
also,
thalidomide can be used for multiple myeloma, and
excellent.
it is called repurposing of thalidomide.
Correct. Very good. Very good. COVID also, hydroxychloroquine we repurposed for COVID, which was used for other malaria and other conditions. So like that, modified claims means here also it becomes repurposed drug, means it is for different indication. Then fixed dose combination. For the first time we are combining, or it was there, now I'm changing the proportion. Sponsor says both are already there, both are proven. I'm just making it 1:4, 2:4. No, the minute you change the proportion, it's a new drug. Then you have a drug, but you are making it sustained release, new drug. Biological products, vaccines, DNA product, rDNA products, monoclonal antibodies, many of them are there, no? Particularly for cancer, for psoriasis, we use these biologicals. So they are all biological products. Forever they remain new drugs because they have a higher risk of adverse effects. Time frame. So any drug which gets licensed, it's in the market for 4 years, it remains to be a new drug. After 4 years, we can consider it to be already approved drug for all except biologicals. They say biologicals will continue to remain throughout their life as new drug. So you should exert lot of uh, caution. And what is the purpose of classifying this? What they say is, well, even if you market, no, this company has to give updated reports. It's called periodic safety update reports to the government every 6 months for first 2 years, every year for next 2 years. Then it can stop. But first 4 years also you are under scan. The company is under scan. Biologicals throughout they should keep updating throughout because they behave erratically because they are biologicals. We don't know the genetics of the person. So these are all this whole thing forms the spectrum of a new drug because we are dealing with new drug development. How are new drugs discovered and developed? If you see the historical development, William Withering discovered digitalis for edema from traditional remedies, isn't it? Reserpine came from Rauwolfia serpentina. Aspirin also, morphine. There are so many drugs from traditional remedies they observed, and from there we have discovered. Then Alexander Fleming discovered penicillin by serendipity, isn't it? He just observed that there was a mold, there was a growth around a culture plate which inhibited some bacterial growth. Then he said this fungus, Penicillium notatum, that could produce something which can work against some infections produced by the fungus for the bacteria, like that serendipity. Ether anesthesia. I am very, very fond of this. William T.G. Morton. You see the book in Bailey and Love, and he is a person who discovered, who observed that ether can produce anesthesia, made a device to administer that in a patient with goiter in Massachusetts Institute. And then they came up with a very good, from then on, the surgery started happening. So that is one. And hydroxy repurposing, as we just said, already drugs are there. Maybe it will help in some other condition. So that is another way of discovering. But here we are concerned about rational drug development. So we all know this, but this need not always happen. Sometimes there is a need. So what initiates, when is the need for rational drug development, right? So existing drugs are not working effectively. For example, now tuberculosis, we are seeing that it is not getting eliminated. Why it is not getting eliminated? There are so many challenges. 6 months of treatment, people don't adhere. They have comorbidities, so many challenges, long duration, and the patients are not so well educated. So is there any need? Can we invent a new drug for tuberculosis? Government is pushing everybody. If any of you are interested and have a aptitude, and if you can do it, please do it, because they're funding. Other bacterial infections, what are the challenges? Our company is working on this because they don't reach adequate concentrations. The plasma concentrations of vancomycin, amikacin, is not as per the therapeutic range because the patient is not healthy. So we need to increase the dose, and that's not happening. So there is increased prevalence and of antimicrobial resistance. And if there is emergence of, so we need a newer drug. Therefore, there are companies, and recently Wockhardt came up with a new combination, isn't it? For it's a beta lactamase inhibitor. They have come up with a new drug. And emergence of new infections such as COVID. We didn't know what to do. Therefore, we had so many repurposed drugs because we didn't know, we were using existing drugs. So rational drug development starts when such a need is identified, or we understand that diabetes or hypertension or any condition is progressing in a different way, it's manifesting in different way. So new biomarkers are there, maybe we should target this. So when such an understanding starts coming up, then the rational process of rational drug development starts. So what is this rational drug discovery and development? We are going to discuss in today's session. Any doubts so far? Anybody? Any doubts? Anybody wants to ask anything so far? Are we okay? Are we okay with this? Yeah. Okay. All right. So now Target identification, isolation and validation of the target, lead discovery and optimization, preclinical studies, and clinical trials. Okay. So, what is target identification? How many of you have, anybody has done this as a seminar? We used to give this as among the attendees today. Anybody has given seminar on this rational drug discovery? You can unmute and answer. Just yes or no. I cannot see the chat box. That's why.
Reverse pharmacology seminar in that target identification had been mentioned, but
Okay.
target identification as such has not been done as seminar, ma'am.
Okay. Okay. Good. So, just to understand, in case you have done, maybe we could. Okay. So, target means a biological target that is responsible for the disease condition, determining which molecular entities, which molecular entity is most relevant. For example, I will just explain this condition. In diabetes, we have insulin as the, pancreas as the target. Pancreas, let's say, there is reduced secretion of insulin. There is also reduced uptake of glucose by the peripheral muscle and the fat. Then they have identified, I have mentioned here actually, then they identified that even there is a SGLT2. We could also keep that as a target and prevent the reabsorption of glucose. Like that, in diabetes, type 2 diabetes itself, different that we could bring about drugs targeting different aspects in the pathophysiology of the disease. Bronchial asthma also. Acute attacks. What do we use? Bronchodilators. Where do they act? They act on the beta 2 receptors in the bronchioles. They just bring about bronchodilation. So the first target identified was beta 2, and inhalers were synthesized. The bronchodilators. Now, before inhalers, it was oral. They said salbutamol like drugs will dilate, and so that is a target. This is a molecule, and fantastically they developed. But they observed adverse effects. Let's explore some more delivery systems. So they went for inhalation, and they went for selective beta 2 agonist. So salbutamol is selective beta 2 agonist. Before that, isoprenaline was used. So like that, it continued based on the target only. That was a target. Beta 2 agonist, beta 2 receptor was the target. But then that was not selective. It was also there elsewhere. Vasodilatation, cardiac, beta 1 and beta 2. Isoprenaline was beta 2. Before that, they used to give adrenaline, which was beta 1, beta 2. So when, I hope all of you are clear, the target they will start investigating in this way, which is a target, which is a drug required, is it selective, and is it most implicated in the condition. So asthma beta 2 worked very well, bronchodilation. Then they found that they understood the inflammation, and then they said if we target inflammation, it will prevent. So that's inflammatory, that those nuclear receptors for steroids, those became the targets, and prevention of attacks was taken care by corticosteroids. So how beautifully there were two targets now, one for bronchodilation, one was for preventing the inflammation. Like that, they will start studying the pathophysiology of the disease, and then initial reports come from the publications. Now AI is being used to collect all the information and to see how the drugs have worked, whether that target is playing a role in that particular disease. So drug development starts from understanding the pathophysiology of the disease completely. When I started working in natural remedies, we were asked to, I started understanding that, as Nanjara said, we did not have such formal bronchial asthma. We were working on bronchial asthma. First time, not even as an MD candidate, when I started working, I understood various steps in pathophysiology of bronchial asthma. And then I understood how the different drugs act at different targets. Antihistaminics was earlier prescribed, now we don't. Antihistaminics on the histamine receptors, bronchodilators, steroids, and then these anti-inflammatory like what are those, disodium chromoglycate and all those, how they work. So different drugs act on different targets. Therefore, the company studies the entire pathophysiology, and then they say which is the most implicated target where I can work. So, and then they will also look at the commercial, and they'll see how, if I block this target, how much improvement it will bring about in the patient's condition. So that decides, that feedback by the physicians later will decide how well this product will be entering the market. So identification of this target. Now let's say we'll go by this only, beta 2 receptors or sulfonylurea receptors. That is, then they will characterize that this receptor, is it a protein, is it an enzyme, is it a DNA? So all this, so for this characterization means understanding this requires genomic studies, proteomic studies, bioinformatics, machine learning, and AI. Genomics is understanding the genetic expression, mutations, polymorphisms, and how expression means if this gene is modified, what will be the outcome? Okay, phenotype it's called. So they'll understand the genotype of those people, asthmatic patients. They will understand the genomic pattern, and then they will see if I block that gene which codes for that receptors, will the condition improve? Or if I enhance the expression, will the condition improve? For example, beta 2 agonists, no, in asthma agonists, not antagonists. So if I enhance beta 2 function, bronchodilation, right? In angina, if I block beta 2, it will stop the cardiac contraction. So what they do is they will take models such as knockout or knock-in. Knock-in is for agonistic, knock-out is for blocking. This is one of the models in vivo. Apart from that, they can isolate the cells, they can isolate the genes, they can isolate DNA, and they can characterize. So genomics is characterizing the gene property. Proteomics is identifying the protein configuration, binding, and all that, because they want to make the drug which suits it, right? They want to make a drug which alters a gene. They want to make a drug which modifies a protein. And bioinformatics is linking entire information which is there in the database. Work that has been done by so many scientists across the world is there in the database. How to link it? That is bioinformatics. And making a meaning out of that is machine learning. You keep feeding this study, this was a receptor, this was a drug, feed that. Then another study, feed that. So they keep, they make an algorithm. If this receptor is blocked by this kind of drug, what is the output? So the machine learns and then it starts predicting. If I make this molecule, what will be my output? Like that is called as machine learning and AI. Any doubts on this? Any doubts? So the same thing, if you want to make an in vivo model, in vivo, then they will identify a particular gene, they'll knock out that, gene. There are methods. I cannot explain because I have not seen, I have not gone so much in depth about the process. We can learn when we do the animal studies. So that is how
Ma'am,
it
can
is.
you briefly tell about the knock-in and knockout? Like, is it that when a gene is deactivated, it is knockout, and when a gene is activated, it is knock-in? Is it like that?
Knock-in is, yes. See, a knock-in is, I will give you a nude mice, they are called. Nude means they don't have some particular genes at all. Okay. In that,
Okay.
nude, they are totally, gene of interest is not there. We'll take that nude mice, which doesn't have, and then we inject a gene.
Okay.
This is what is my understanding. Knock-in is conferring extra properties by giving extra genetic properties to that animal. That is knock-in.
Thank you, ma'am.
Knock-out is removing that.
Okay.
Removing that, and if you have seen CRISPR, it is called gene editing. There is something called CRISPR.
Yes,
What they
sickle cell anemia, they used valine and glutamate, they changed it.
Yes, isn't it? So like that, they will remove that portion of that part of the gene which encodes for a particular enzyme, and see whether the disease worsens or disease increase, because that is a target for us. If we have identified a particular gene, whether whether what happens to the phenotype, how does the animal behave? Does it manifest that disease? Like that, So
Thank you so much,
that
ma'am.
is knock-in and knockout, right?
Okay.
So that is what. Now, having known this, there is combinatorial chemistry where millions of different structurally similar virtual compounds, chemicals are synthesized and their structures are identified and stored in the computers, and one part of that, that they take. Once the target is identified, structure of target is known, they will start studying the combination of this target protein with that chemical which we have already synthesized, which is there on the computer. So they do a virtual combination. We are having next session on docking studies, that is in silico drug receptor interaction. We are going to see that in the next session. So they have this library. Dynamic library means they will have different types of compounds which are complementing also. Maybe individually you can target or you can combine like this. So that if the target is of this shape, just a schematic representation, if you cannot target with this or you cannot target with this alone, say they combine, and then these are called as high affinity ligand. Ligand, see here, you can see how it has bound like a lock and key. So they make such combinations virtually, on the computer. This is a very advanced computational modeling because they don't want to waste time and spend money on the biological systems. They will do everything on the computer, and they come to, it's not the specific drug, they come to some conclusion. Some few hundred molecules, 1,000 molecules, I should say, are able to combine, and there are parameters which will say what is the affinity. Affinity means the ability of the ligand to combine with the receptor, right? Okay, so that the ability of the ligand to get attracted and combine, because it's complementary in configuration, isn't it? So it will go and combine there. So this is high affinity. They will choose these high affinity ligands as a first screening, and then target validation. So there also we said knock-in, knockout continues here, because if for target identification also, in validation, the knockout mice, knockout or knock-in, now they try with the new molecules which they have synthesized by combinatorial chemistry, and in silico models, whatever they have identified, now those are tested on cell lines or in the models. So that confirms that that is a target. Validation means reproduction, proving that it is going to work. That is called validation. So this becomes the next step. Studies that are performed to confirm that these are the targets actually involved in the disease process by manipulating the target with the drugs. So this is called target validation. It can be done in vitro, that is outside the body, using cell lines and cell strains, or it can be done in the animals also. Then very interestingly, now they have so many compounds virtually as well as here. They would have had, usually it runs in hundreds to thousands. Now they will run through proper computational as well as in vitro in the lab. They will run in small ice in vitro screening, it is called. Again, some cell lines for pharmacokinetics, for pharmacodynamics, for toxicity, for target binding. So these are all on the wet lab as well as on the computer. And wet lab they can do in the cells or in the animals, and both they can use such sophisticated equipment such as MRI, X-ray, crystallography. They can visualize what is happening using this technology. They will visualize whatever targets we have designed, the best, I told you, high affinity ligands. Are they really combining? That can be again confirmed using this high-end imaging technologies. And not only the combination, they will start working on whether it crosses the membrane, lipid solubility, how much crosses, because that will give us an idea of bioavailability, isn't it? And whether it is damaging the cells. They take the that's called MTT assays, cell viability assays. At what concentration it is damaging? Are they having toxic properties at a very low concentration? Then they abandon that. Ligand binding is good, but it's causing cell damage. So all these will be studied step by step again. So among these, they will identify out of so many molecules, they will find the best molecule, that is called lead. From hits to lead. Hits were preliminary screened molecules. Lead is maybe top 10, you can say, top 10. Then how do they arrive at this lead? I told you they're called high throughput screening. High throughput means in a day they can screen thousands of molecules, 10,000s of molecules. AstraZeneca was one of the company those days when we were working in natural remedies. Those days, 25 years back, they had this high throughputs because they were into proper R&D. So there they use automated testing of large number of compounds for a specific biological target in assays. So all this data, it will be stored, and that many of the companies are using this available data for AI now, because that can predict. This will really give them good amount of data. Whenever a new molecule is added, it will check the compare the structure, and then it can predict to what an extent this can be useful. So this is how they are working. And hit rates are 0.5 to 1%. That means if you screen 100 molecules, only one will qualify the criteria which they have put. I hope I'm clear, all of you. And in this, these are all, what are these? These are all small tubes or plates, and 100 small, they contain the, let's say there's an enzyme. Okay, there's an enzyme, there is a substrate, and your new drug. So whether the new drug will, enzyme will act on the substrate and produce something. Xanthine oxidase, let us say. Allopurinol acts on the xanthine oxidase and inhibits uric acid, isn't it? Am I right or wrong? Anybody? Something like that. Okay, I just picked up one, this one. Or alpha amylase. It is an enzyme which acts on the starch and breaks down into monosaccharides. Starch is broken down to monosaccharides. So if you have alpha amylase, you have starch. If this drug prevents the breakdown of starch, then it prevents absorption, isn't it? Like your, which is that drug which prevents your absorption of acarbose, isn't it? Which prevents the alpha glucosidase, alpha amylase, like that, acarbose, anti-diabetic drug. So if you will put the enzyme, you will put the substrate, and then add the drug and see whether enzyme action is prevented or what, whether it prevents the enzyme. So starch remains as starch, right? Starch remains as starch, it gets excreted. The in vitro we cannot study, but it will test the effects of enzymes. Or if there is a cell, anti-cancer, you have a cell which proliferates and you add the drug, it inhibits the proliferation. Such cell lines also can be tested using high throughput studies. That means everything, there will be 1,000 different molecules, and all the tubes will have the same assay system. Reaction will be the same, but the drugs will be different, So out of that, and in different concentration, of course. So it will pick up, it will be programmed in such a way, it will pick up the best 100 maybe. So like that, high throughput means more number of studies can be done. Cell lines, enzyme assays, etc. Do you know of any example where we can use a whole animal and have high throughput? Can you use so many rats for screening? Can you put so many just like this? No, give injection to one rat, one mice, hundreds of rats and mice. That would be so unethical. But we have come out with alternatives. Anybody is aware of such alternatives where whole animal is used and we get high throughput screening? Anybody? Whole animal, mammals, not mammal, mammals only. Yeah. Have you heard of this? Okay, so these are zebrafish and Drosophila, housefly, right? For high throughput screening. So Drosophila, there's very small, fish, zebrafish, brine shrimps. They have come out with that now. So these actually, if you inject a drug, you can see there are so many training programs. Some of your seniors have attended. I think Mallikarjun, did you attend zebrafish training? Are you there, Mallikarjun?
Yes, ma'am. I have attended.
You attended, right?
Yes, ma'am.
These animals, I mean, these zebra fishes can be injected, and see here, so many well plates, just like enzyme assays, no, you can put them in these plates and screen large number of molecules using these zebra fish. This is very useful alternative model for animal disease. Now we did lead target identification, target validation, lead identification from hits and through high throughput screening and zebrafish and others. Now we have to optimize. Now we have got 10 or 15. Now we have to optimize. Optimize means what we will do is the structure is known, right? So this is the active site, and this is the compound. It combines, but it is not fitting properly. So I will make a little modification and see that it fits properly. How do I do this? Every time I modify, I subject it to some studies. I modify and do, modify, come back, modify, come back, until I reach the appropriate optimized molecule. Lead means it's a drug molecule only, until I reach. So I pick up the best 3 or 4 leads by modifying them, putting them in the relevant experimental models. So one of the criteria, this is just, there are so many, I didn't go into that. There are some criteria for picking up the right drug. I did not want to go into that depth, but this will be asked for you in examination. What is Lipinski's rule of 5? In the very early stage, at the stage of chemistry itself, they define this, Lipinski has defined for oral drugs, molecular weight should be less than 500 Daltons. H-bond donor should be less 5. H-bond acceptor should be less than 10. H-bond acceptor should be less than 10. Lipophilicity log P should be less than 5. So these are all defined chemical, this is liquid partition coefficient. This is lipophilicity. How well it partitions across the cell membrane, that they have in vitro models. Based on that, they will decide. So that completes our, discovery, design and discovery. So we have discussed target identification, find a biological target such as protein, that is receptor or nucleic acid element with activity that a drug can regulate. Validation, demonstrate, confirm the target is involved, modulate, knock-in, knockout, that the target may be beneficial, modulating the target may be beneficial. Lead identification, find a compound that shows desired biological or pharmacological activity, may initiate the development of new clinically relevant compound. Lead optimization, improve the effectiveness, reduce toxicity, and increase the absorption of the leads. As we said, refinement in pharmacodynamics, absorption, distribution, metabolism, excretion, in vitro, in vivo, both are done. These are part of lead optimization. So that much we have done. And from here we move on to, so we have completed this, we move on to preclinical studies. Okay, so far anything, any doubts? Any doubts, or anybody would like to add anything? Anybody? Please feel free. Any questions? Do you think, this is an open question, Dr. Harshad? I think I'll come back to you. Harshad, are you there?
Yes, ma'am.
Yes. Dr. Harshad, do you think these are relevant for us considering our career opportunities? No, we discussed about some have joined very recent, just now. Many are just still joining. I could hear that. So we told our career opportunities are in medical affairs and in pharmacovigilance and in R&D, academic, medical writing, and what else? So these were some of the career opportunities. So understanding these, does it help in any of these career activities? What is your opinion, yours and others? First, I will ask. Do you think we should know this as pharmacologists, whatever we are discussing now?
Yes, sir. Yes,
As a
definitely,
pharmacologist.
yeah, definitely it will have ramifications across all of the career options.
Yes.
So in medical writing also it will help.
Yes.
Writing the reports, even in
Yes.
academic research, all these clinical trials and
Yes.
how to sort the research also. And even in, of course, in new drug development, pharmacovigilance, everywhere it will have these applications for each
Application. Yes. Right. So supposing, as he said, you are doing some herbal product, because most of us do that. It's easier, rather than, so we can validate, these are all established methods. So you have a herbal extract. Why don't we, to validate that, to say that, let's say we did one study on Syzygium cumini, jambulum seed extract, ನೇರಳೆ ಹಣ್ಣು ಕನ್ನಡದಲ್ಲಿ. So that was tested on cell lines. Cell lines is a accepted model. We had insulinoma cell. We purchased that, that cell line. We saw the effect of that extract on at different concentration in vitro, how much insulin it released. It was doable. I can share that actually in one of the presentations. I will share that. We did that in collaboration with an R&D lab. He had all the setup. We took our extract and he helped us. We put that extract, incubated, and in the supernatant, we took the aspirated few microliters and saw in control without the drug what was the insulin level. In the treated, the drug administered Petri plates, what was the insulin level. It went for a good publication. Very simple. Now, of course, we spent a little. The girl was doing PhD. So these are all established methods which an academic researcher can do. And when you read the protocols in clinical trials, they would have described all these. This product was tested in cell lines. This product showed in zebrafish. So in CRO or medical affairs, you are sitting with this document, you will be able to appreciate, okay, this is how it was done, zebra fish, and this is how, because I'm exposed to zebra fish model. So that is how these things will help you to interpret and understand and get connected. Otherwise, it becomes Greek and Latin. All the documentation becomes Greek and Latin. Clinical trial contains investigator's brochure, which has the entire history of the product, new drug molecule. So new molecule. So there they would have described all this. So you should able to understand. All the while, no, we used to teach only animal studies and clinical trials. Now, as a curriculum also, we are going little behind what happens, because, and this is generally, these are done by biotechnologists, pharma industry people, pharmacy graduates, life scientists. They do beautiful research because they're also trained in that way. No harm in pharmacologists collaborating with these people and together developing a molecule, or together understanding repurposed drugs, we can further evaluate. Anyway, now I'll come to preclinical studies.
Ma'am, in between, can I ask a small doubt, ma'am?
Please.
Okay, ma'am, I got disconnected in between after a lead target validation and target optimization. Between I lost in between, but it's okay. Okay, no, after a
No,
target
okay, target
validation,
identified
yes.
and validated.
Then target validation. Then when I rejoined, I couldn't rejoin from my same account, so I had to connect via another account. So then I reached target optimization. So in between, if I missed something,
Yeah.
I'm so
No,
sorry to disturb.
No, that's okay. That's okay. I will repeat that. But also, I want to mention, I will put this on YouTube. Again, you can go through also. But
Thank
right
you so
now,
much. Then it's fine, ma'am.
that's okay. No, that's okay. You can follow others also. Others also maybe. So target we identified, we validated. Then
Yes.
the target, in target validation, we had molecules already. That's how we validated. No, now that we will go for lead. We have so many molecules now. We have the target. Now I have one big chunk of molecules. I will start screening them one by one in the same target. That target is put in this petri, in these plates. Plates means in the lab there is a petri plate or there is a tube. In that we put the biological system on which we want to test the molecules. As I said, enzymes we want to test. Enzyme assay, enzyme functioning. Substrate is there, enzyme is there. It will produce the end product. That is the assay. It's called as assay. There I put drug, different drugs. Same target. In all 1,000 assay method, assay system, same target is there. But the drug differs. 1,000 different molecules, slight variation in the their structure, their functional groups and all that. So let me see which of them works best. That is how high throughput screening is done. And we also do in zebra fish and Drosophila, similar thing, high throughput, but it's a full animal, it's not a mammal, of course, it's a fish. But we take that, and because it's a small in size and there are plates, we can screen number of molecules in a day. And then from there we refine, out of those 1,000, we come to 100, pick up the best 10 and see which works best, keep modifying back and forth. And then we reach the final drug. And then there's Lipinski rule of 5, which will guide us chemically how best we can pick up the best drug. So I have repeated this. And then now we have...
So far we were talking about the targets in the targets of disease targets. And ma'am, another doubt I have, there are a few traditional medicines, like as you told, we must collaborate everything. And as you know, the famous nootropic Brahmi. So, and it has it has been used in various Ayurvedic preparations and all, and even clinically it has been, there was a session in PJ Chandigarh Ayush conference, and so there was presented so many, even psychiatric drugs etc. has been developed, and for alchemists also, but it is very difficult to identify which particular molecule. Brahmi has lot of molecules. So what we discussed today is identifying the target of the disease, but how to identify, if a given plant or given medicine, which molecule of that medicine is useful for that, because they use the whole the plant.
So there is no standard protocol there. In herbal, it reverse pharmacology works. They start from first they screen in vivo, and then they come back to demonstrate mechanism of action and to identify which molecule is working, isn't it? That's what you're asking. Brahmi is useful. Which of them? one lead I can just give based on my ideas. If you have 20 of those, you there also, it's not very specific. In herbal, this is the biggest challenge. They will have flavonoids, they will have gallic acid,
Yes.
allergic, so many things are there. You can test all of them in such in vitro models, but this is a big discussion actually. Your good name, I forgot.
Dr. Revati Benjamin.
Revati.
Dr.
Ha,
Revati.
Revati. So, Dr. Revati, what happens is in herbal, they don't act in isolation. They act synergistically. So, the method of drug discovery in herbal is not exactly the same in rational drug design.
Okay, fine.
There, you will have to identify which is a predominant molecule which is contributing. But you isolate that and give it to the animal, it will not perform similar to the whole extract. Because
Yeah.
whole extract, this is my experience because I worked in natural remedies, it's a combination, synergistic activity of multiple molecules which will bring about the effect. This is what we have heard. So we can discuss that. We have a session on herbal drug development. I have
Thank you
got
so
a
much.
person who is vice president of that company who are involved in exactly what you have asked. So we'll come, that's a very specific question. We'll come to that definitely, but that is a challenge. But we can apply these principles and see how best it suits the herbal drug, right? We can
Okay.
do that. Okay. Now
Thank
coming
you,
to
ma'am.
preclinical studies. Preclinical doesn't necessarily mean animal studies alone. There are many more. We are almost through the presentation. Then we take up questions. So preclinical is stage before clinical trials. So preclinical animal studies are to determine a drug's efficacy and ADME, absorption, distribution, metabolism, excretion, and toxicity. All these things are estimated in using animals. And the maximum, I mean, the most useful aspect of preclinical is we will see what is the maximum dose these animals can tolerate. From there, we extrapolate to first human dose. We will see what is the maximum tolerated dose. More detailed discussions will come when we have those sessions, particularly. What happens is animals are very close to the humans, and because rats and mice, particularly in terms of physiology, not higher functions. Higher functions we use monkeys, chimpanzees, and all that. Otherwise, we use rats, mice, hamsters, guinea pigs. So that dose which starts showing the toxicity, there is a method how to estimate this toxicity, which organs to be harvested, which organs histopathology to be done, how to assess the behavior. So there is a protocol for that, internationally accepted protocol. We follow that and we see which is the maximum tolerated dose. From there, we calculate the first human dose. It will be usually extrapolated human dose, 1/10 of that, extrapolated human dose, and then we start 1/10 of that. So there is a way to calculate. That is for toxicity. Efficacy, we have so many screening models actually. We can induce a disease, or there are some spontaneous models. I'm sure all of you will be writing answers to this in your examinations. What are the methods available for screening anti-diabetic drug, antihypertensive drugs? So this is a general description. You can have spontaneous, genetically modified, knockout, negative model, as we said. Healthy animals we can induce, biologically induced, lesion induced. So like that, different methods are there for depending on the therapeutic area and the drug. And then we use all these animals for different types of toxicity studies. Probably you have not heard that we have done a toxicity study. When we do acute toxicity, we collect 5 organs for histopathology. When we do chronic subchronic actually, we collect 27 organs, 27 organs, the liver, the kidney, the eyes, this pituitary. You'll have to not 27, 28 organs are collected from the animals. So that much of in-depth toxicity is done. So by the, we learn this more in the further sessions, those who have joined. Others can refer to OECD guidelines, Organization for Economic Cooperation and Development. Those are the guidelines which every pharmacologist must know, to interpret a protocol, to understand the product. Maybe sometimes the company, with due respect to the sponsors, generally they don't do that, but they might have overlooked. As a pharmacologist, if you point out that this toxicity level is not acceptable, or we should observe for this ADR, go and talk to the clinicians as a medical affairs person. Preclinical studies have shown this kind of toxicity, which was tolerable, but we may get this ADR. So be watchful. So these are your original ideas, which you can get if you understand the animal toxicity. So that is about animal, and then we study the absorption, bioavailability, all that. And then others are formulation, that time till the lead optimization, we had the only molecule, pure compound. Now we start administering, no, first human dose we should give as tablet. So they will start doing the formulation, which excipients to be used, what proportion it should be used, and how do we administer, tablet, capsule, powder, syrup, or injectable. What should be the route and what should be the dosage form, and then they do the stability testing. In the meanwhile, how long it is stable, all this will be done. These are the animals, and then there are so many alternatives to animal studies. We have a session on that also, but very well evolved. And the best of that which I liked was organ on chip. They have liver on a chip, microfluidics. This is one of the areas where we are also working, but not animal studies. We are working on something else. They put the cells here, liver cells, and they put the drug also. And then what elutes is a reaction between the cells and the metabolites, rather. They put the liver tissue is already embedded in the microfluidics miniature, or it's an organ on a chip. And then they put the drugs, and then they see what happens there. Organoids are there. Cell culture is there. Fibroblasts are there, so stem cells. So like that, a lot of developments have happened as alternatives to animal studies. They also use brine shrimps, the small fishes. I have seen this in natural remedies. And then zebrafish, we have talked, roundworm they use, and others, many things are under evaluation. So these are also things that can be used. Now coming to the last one, what are the regulatory requirement and ethical considerations? Okay. Studies to be conducted in labs that follow GLP, good laboratory practices. That means, have you all heard of NABH, NABL in the hospital?
Yes,
Please,
ma'am. Yes.
you have
National
heard.
accreditation.
Yes, Revati, you have heard. What about others?
I worked in General Hospital Ernakulam in Kerala, so there
Okay.
we had NABH accreditation.
Yes.
So
Anybody else?
Yes, ma'am.
You
Our
have heard?
hospital is NABH accredited.
Very good. Anybody else?
Yes,
Only
ma'am.
yours
Even
also?
our college,
Yeah.
Oxford Medical College, has it.
Yes. Good. Whether your hospital is accredited or not, you should know that there is something called NABL, NABH. And likewise, for toxicity studies, there is something called good GLP accredited laboratory. That will be accredited by GLP, International Accrediting Council it is. Then we have committee for, have you heard about this? All postgraduates should know that central government has made a committee for control and supervision of experiments on animals. That was initially control CPCSCA. Now it is called CCSCA. Right. And now institutional animal ethics committee approval should be taken. And institutional biosafety committee approval as required. Good manufacturing practices for manufacturing that molecule should be there. And after this, the sponsor, after this lead optimization, the preclinical studies, formulation, stability, he applies for clinical trials. Anybody? Any questions? Any questions? Anything so far?
all good, ma'am.
Yeah, okay. So then clinical trials, many of you know, clinical trials progresses in 4 phases. Before that, there's phase 0. I'll tell about that later. In phase 1, so let me go to that first. Phase 0, they do first. Primary application. First time we have come out, we have done animal studies. How can I administer? If any one death happens, then he is accountable. So he will do microdosing, very small dose. First human dose they have calculated, but in that also they will take very small dose, and they will see whether it gets absorbed. It comes in the blood. They do estimation using HPLC, LCMS, and then see it's less than 1/100th of the dose of the test substance calculated to yield pharmacological effect with a maximum dose of less than 100 micrograms, not even milligram. 1,000 micrograms make 1 milligram. So they are using 100 micrograms maximum. If it is less than that, whichever is lesser. So that they will do. Once they are sure of it gets absorbed, bioavailability and all, they'll go to phase 1, 20 to 80 participants up to several months. Studies the safety of medication. And usually 3 or 4 molecules come here and 70% success rates. That means not all go through. They may come back, modify the molecule, again come back. That happens. like lead optimization, here also they may go back again, then do again some studies toxicity and come back. Phase 2 will be for 2 years, studies the efficacy in the patients, healthy human volunteers in phase 1, except for cancer and HIV. And in phase 2, they will study the efficacy in 100 to 300 participants in patients. And again, here, not sure, 33% success rates. Once again, they may have to go back. Phase 3, some more. And here also they may get, but with by then, actually little more they would have modified. And here they study multicentric. They studies multicentric across different countries, different states. They do that for different patients in different ethnic background. Once they do that, they submit for approval, they get marketing approval, and then it goes to the market, but under observation. Long-term adverse effects, pharmacovigilance, post-marketing surveillance, all these are done after it is marketed. At every stage, there is approval required. Before phase 1, approval by the government. After phase 1, submit the data, get approval for phase 2. Submit the data, phase 3. Submit the data, get marketing approval. Keep reporting whatever you get to the government. So here, preclinical data, then they file the investigational new drug filing, then investigational new drug approval government gives. Phase 1 you do. Then again decision is taken whether I should go ahead or not. If he goes ahead, submit, again get the approval, New drug application, investigational new drug, this is new drug application is filed. So here it becomes a new drug, marketing permission, and then it goes for phase 4. So are we clear about this? So regulatory approval is not required by the government for animal studies, but it is definitely required to conduct the phase 1. Before phase 1, before phase 2, before phase 3, and during phase 4. For first 4 years, it is required. For drugs, throughout it is required for biologicals. This you must remember. Guidelines for conducting, we have international ICH GCP. We'll be discussing about that. We have new drug clinical trial rules, ICMR guidelines, and institutional ethics committee approval as per their SOP, standard operating procedures. You should know all this because when you face interviews, many corporate heads say that PGs are not aware of the terminologies, SOP, GCP, GMP, GLP, you should know all this. Then what are bioavailability? We discussed this, no? This is for new drug. Then we said generic and branded generic. So these are Me Too drugs. The patent expires, then some Indian company produces Calpol, Rosin, Calpol. So both are same, different brands they are, isn't it? Or paracetamol itself may be produced by Reddy's, Dr. Reddy's, we told that. So how does it compare with the innovator drug? We do bioavailability, bioequivalence studies. Drug is administered to the patients who are hospitalized. These are clinical trials used to demonstrate that generic is a not innovator drug, Me Too drug. It is therapeutically equivalent and biologically equal, not therapeutically equivalent to an innovator. We cannot do studies, clinical trials. So we measure the bioavailability. We need not do it in patients. We will do it healthy human volunteers and we see how much of bioavailability is there, and that will infer it will be effective as the innovator. So we administer the control innovator drug, we administer our generic drug, admit the patients for 24 to 48 hours. You are going to visit this setup. That is why I said it's very interesting. So there you collect the samples and then they do it. And then if you find it doesn't vary, plus or minus 20%, then the regulatory authority says you can go ahead and market your generic product. This is BAB studies. India has many BAB setups and lot of generic drugs manufacturers we have. So drug development is a multidisciplinary effort. It starts from early drug discovery. We have discussed all these steps: target identification, hit discovery, screening, high throughput screening, hit to lead optimization. Then we do assays, in vitro, in vivo, ADME, proof of concept means animal models of hypertension, diabetes, etc. How the drug can be administered, formulation, dose range, IND application. Then we do clinical development, phase 1, healthy volunteers, phase 2, phase 3 patients, dose escalation in all these, safety, efficacy, pharmacokinetics. Very important is method, bioanalytical method to measure the drugs in the plasma. Our company is working in this area, and FDA will review, FDA or CDSU, sorry. That's why I put CDSU or FDA. FDA is for American Food and Drug Administration, USA, regulatory authority is called FDA, and they review this, and ANDA is abbreviated NDA for generic drugs, abbreviated generic drugs, and then we have post-marketing by CDSCO. Now, few terminologies and then we wind up. Post-marketing surveillance, it is ongoing monitoring of drug or medical device safety and effectiveness once it reaches the general public. It's now free. When we did clinical trial, we did under controlled conditions. Now it is going free. It collects real-world data to identify rare or long-term side effects that were not detected during smaller pre-approval clinical trials. All right. So now post-marketing surveillance, particularly for first 4 years. Now the postgraduate thesis or the faculty projects, many people take up this one. It's really wonderful to see the new indications, or it's a new drug. Okay, new indications also becomes a new drug. 4 years also it's a new drug. You can watch, the drug is introduced. Let us see in our area with comorbidities, because the company says you can give this anti-diabetic drug even in hypertension. It has proven. So let us study what happens, because some physician may say, I found a drug interaction. Then the pharmacologist will join the physician. Let us do some more studies, sir. Let us take this up as a project. Then our postgraduates are there. Can we give this topic for their thesis? Because already the drug is in the market. So it is safe to do actually. Such studies, drug utilization, how many physicians are prescribing, how the patients are feeling. This you can do independently as a thesis project or academic project. And if you are already working in the sponsor company, you can tie up with these people and then generate a lot of data for your company. Medical affairs person can think of such if you are in that area. This will add value, collecting a lot of data on the drug which is just marketed. Semaglutide, they are running left, right, center to collect data now, because it's introduced as an anti-obesity also, isn't it? So like that, right? Then what is pharmacovigilance? You know very well. Detection, evaluation, understanding, and prevention of adverse drug reactions. And in the industry, there is collection. In the hospitals, there's collection and reporting. And the hospitals may call up, the persons may call up the companies directly. In the abroad, it is like that. And here individuals can call the PVPI. There is a call center there. Number is there. You can call. Then companies will do aggregate reporting, signal detection, assessment and monitoring. They will have risk management. These are all the areas where you will join as a MD pharmacologist. Very interesting area where you will contribute to this, whether the drug is causing an ADR or not. Large number of data is available. Real-world study evaluates how medical treatments and interventions perform in routine clinical practice, similar to post-marketing surveillance, but now this area has evolved in a big way, because no sponsor wants to lose his molecule. So this was not there about a decade ago. Now it's picked up in a big way. Lot of data is being gathered, lot of data management is being done. So in clinical trials, it was tightly controlled. Clinical practice, you give it freely, something may happen, and or maybe you may get additional beneficial effects. All that constitutes the real world data from every patient care. These help the researchers to understand treatment's long-term effectiveness, safety, true to life outcomes. Now lastly, I come to precision medicine. So precision medicine is where we all the while, listen to my words, then you can see the picture. All the while it was one treatment, majority, any drug you give, we assume that all patients will respond, knowing very well even during the research only 60 to 70% response is there, 30% don't respond, but we market it as a sponsor. When the physician starts prescribing, he will say this patient has not responded to atenolol as an antihypertensive. He will increase the dose. He'll say, no, he's not right. Let me change the drug. What made that person not respond to atenolol? What made the person not respond to metformin? So these are not studied. Now, with the availability of genomics, therapeutic drug monitoring, AI, they don't want to lose on that product. The sponsor will feel my atenolol has gone, he has moved on to some metoprolol. Why I should lose it? Let me define who responds to atenolol. Then what he will do is, when you prescribe, he will give, these are the patients who respond to atenolol. Don't prescribe if it's not there. But it's a long way to go, because these are the patients means They are giving genomic characteristics. They're coming out with kits which will define who responds to which drug. I hope I'm clear. So what happens is when you give a drug, it is not going to be effective in all because we differ in our genetics, in our other structure. We have metabolomics, we respond in a different way, diet, everything. So they had done one study on DMARDs, that is rheumatoid arthritis. So what they had done is they found that they some have good response, some have inadequate response, some have no response. So likewise, they categorized by risk, by their diet, by their multi-omics, genomics, proteomics. Then they came up with different types of drugs for this. They come out, rather, they have not come out, they'll come out. Or you can also alter the dose, which we are doing now. The same dose also, they may differ not only in the response is not only dependent on the drug. That dose may not be efficient because there may be rapid acetylators, slow acetylators for isoniazid. So that will decide how much dose is required. So precision medicine is where we stratify the patients into groups and see what works best for whom based on their characteristics. So I must tell you a very interesting thing. I have heard and I know that person IASC, they are collecting data from TB patients, collecting blood samples, and they are doing a lot of these omics, and they define what is they define what is called like this genomics, transcriptomics, all these, they're called signatures. Every person has a signature, they say. This is a very high-end research. If you can understand, fine, otherwise leave it. But this is how the research is progressing. This is how pharmacology is growing. We should be aware. So precision medicine, patient population will be mixed like this. The standard approaches, you give treatment A effective in 20% of target population, 80% may be waste. Now they will define tailored approach. They will categorize them and then they will define treatment A, treatment B, treatment C, treatment. They'll modify the product just like we did lead optimization. Like that they will do. That is called precision medicine. For precision medicine, we need genomics, we need metabolomics, we need proteomics of structure, in a person. They'll collect blood and they will do all this and they'll find they have found so much of variation. They are working on this. Very expensive. It's not in practice. It will come. But we are trying to make some, we are entering precision medicine. We'll tell you how it is. So if you become an academic researcher and innovator, you can contribute to real world study. Drug utilization is one such real world study. You can initiate an investigator initiated trial. Different dose of existing maybe pre-anesthetic medications. Many postgraduates will do that. You can do a collaborative drug discovery, device innovation in a collaborative way, AI tool development, excellent area actually. You people will do very well if you collaborate, right? So what we are doing is precision dosing in tuberculosis to prevent therapeutic failure, precision dosing in sepsis to prevent AMR to antibiotics, precision dosing in epilepsy. We are developing some tools in therapeutic drug monitoring, pharmacogenomics, artificial intelligence. So this is what we are doing. This is what has been my journey. And coming back to okay. Mallikarjun, can you tell me a specific question? I saw a message. Can you unmute, Mallikarjun?
Nothing.
Google form, I think you're referring to that, right? Okay. All right. So this is what we are doing. And we have collaboration. Without collaboration, nothing will happen. That is why I said multidisciplinary. Make friends. If you want to develop in pharmacology, get network to people, interact with everybody across all cadres, across departments in the hospital, and outside with pharmacy people. They do good formulations. Pharmacy professors, very good in formulations. Damn good, I tell you. So we must learn lots from them. And industry people, manufacturing people, all these people you have to interact with and you can collaborate. And that's how we have collaborated with so many organizations and learned so many things, biotechnologists, clinicians, oncologists. So I have got clinician friends are helping me.
Ma'am, can I ask you one more doubt if you don't mind?
Please.
Ma'am, I saw that your area specialization also include that prevention of epilepsy and failure studies and all, precision dosing in epilepsy. So,
Yeah.
ma'am, recently I saw a pediatric case of Lennox-Gastaut syndrome. I don't know because
Ah,
they even went to AIMS, Bhopal and all, and the child is having a specific drop attacks with
okay.
staring seizures, and the child is on levetiracetam, midazolam, and child is just 13-year-old, and
Yeah.
she had an history of birth asphyxia, which led to cerebral palsy, and she's a little bit difficult to walk. But
Okay.
epilepsy started only 1 year before. So
Okay.
usually in Lennox-Gastaut syndrome, we see between 2 to 8 years it happens. But can it happen so far? It had not been like this. Like she hadn't had any epileptic seizures, but just
Okay.
1 year before, and all the classic symptoms of Lennox, like cerebral palsy with delayed onset seizures, drop
Okay.
attacks, everything is there. And her sleep
Okay.
EEG was not done, but
Okay.
she has multiple seizure types, clonic, tonic-clonic, and absence and everything. Sodium valproate was tried; it is not effective. Midazolam is not effective. Levetiracetam, clobazepam, nothing. She's experienced it almost daily, and
Okay.
um, so they were asking me like I had some experience working under DMHP as medical officer, district mental health program in Kerala. So we used to prescribe neurology drugs also because neurologist was not available. Okay, but unfortunately in government hospitals, still carbamazepine and all such drugs, newer drugs are not being available. So, ma'am, this is so in such cases, like anything regarding Lennox-Gastaut syndrome, where multiple drug failing, epilepsy. So anything so far?
Yeah. Our approach is not we have just started. Our hypothesis is the same dose is not going to work in all individuals, and there's a lot of
Yes.
literature to support. More than in one study, they have shown more than 50% of the patients showed lower levels of phenytoin. Levetiracetam, we
Okay.
have not phenytoin. So we believe therapeutic drug monitoring will be a first step to screen whether it is reaching in adequate concentrations or not. Then
Yes.
investigate what is the reason for that. If it is not, then genomics also we can do. If it is not genomics, there may be other factors which are preventing. One of them would be malabsorption, simple malabsorption, which will
Okay.
not absorb any of the drugs. So if you
Okay.
do that, you will come to know. This is our hypothesis. We
Okay,
are
ma'am,
working.
is this precision medicine currently only in textbook the same? Because our aim is that everything should reach up to patient level.
No,
So,
it is happening. It's happening. No, it is happening because Infectious Disease Association worldwide, they have recommended, and in Apollo, they are doing precision dosing of vancomycin is being done. They estimate the trough level. KEM, there's a publication. Very few centers are doing.
so
KEM
currently
hospital,
it is confined to just antibiotics and not to other drugs.
not to other drugs, because we don't have facility for TDM. TDM is one of the approach. People are not convinced because we're not able to do. So we are developing a tool, and hopefully if it's successful and many more tools come, then it becomes easy for people to monitor the blood levels, bioavailability, and administer.
Thank you so much, ma'am, and thank you for
Thank you, sir.
clearing all the doubts. It was a very excellent session. Thank you so
Yeah,
much.
thank you. Some more, one or two more slides are there. So based on this, we have formulated this course, 6 months. It will start from, we started from today. This was a first session, and we have planned for 6 complete months, and it's open for anybody, I mean, who is interested. I said last time one guy was PUC2, about to join B Pharma from Gujarat. He learned it and he joined. He was attentive. So anybody can join, but we have given, there should be some background of interest and background and commitment to attend all sessions. It's not just register, pay money, and remain absent. That will not work, and we won't be happy with that. We are covering all these topics. If you want to make best use of that, you can join. You can still, there is scope to register. Excellent faculty I have this time. 2 more yet to be added. One is Dr. Ponnagai from Tagore Medical College, Tamil Nadu. And one more is Dr. I want to have, I will not mention. He works in a US university, MD, PhD pharmacology. He's doing very good academic research, and I hope he will join us and share how MD students who are interested to go abroad, MD or M.Pharma PhD in M.Pharma biotechnologists who want to work in pharmacology can get access to international universities, how they should prepare themselves. I'm very keen on getting him. We have all these faculty. So thank you very much for your patient hearing. I hope all of you are there and have not slept and have not dropped out. So open
Ma'am, what
for
is the course fees and details?
course fee. We are charging 12,000 for 6 months. And
Okay.
if you are more than 5 students, then we can we will bring it to 10,000. And uh, in fact, some students, I'll tell it openly, they could not pay in one go. We have given
Yes,
installments.
ma'am.
Installments,
Mhm.
3 installments we will give. But
Yes, ma'am.
the highlight is we will take you to industry, introduce you to Syngene. They have agreed already this year also, Natural Remedies, Syngene, and Apotex, and another bioanalytical lab. And then we'll have protocol writing and CRF designing, ICF in offline, one full day. That's going to be hands-on workshop. These are the 2 additions, and I've added herbal drug development, AI, and device development. These are the 3
Ma'am, to
additions.
be honest, I have always been a fan of this traditional medicines, and I always wanted, like, when we study during my second year, Tripathi itself, it has been written that we have someone called Adal Odaga. So in that, in that cough syrup component,
Ah,
so
you're Ayurvedic person. No, you're MD
no, I'm
pharmacology, right?
MBBS and MD pharmacology, MBBS after MD
Okay.
pharmacology. But like I've been
Okay.
interested in various things. I've been reading about this, and
Yeah,
somehow
yeah, it is
I
good
like.
area. Yeah.
Yes, ma'am. So I had been thinking about this. So like I had been planning, like, ma'am, is this a good option to take DM clinical pharmacology? Does it develop, like, help us in drug development, or are there any, like? Can you just please guide regarding that?
DM, not
Because
DM, no, DM in clinical pharmacology for herbal, you're saying?
not for herbal, for drug development. Like if one is journey, like the main reason I took pharmacology was just because of drug development. I was very much interested in drug development, and I know, like, many plant sources, like everything. It is like, even traditionally, like, I am from Kerala, so in Kerala, rich culture about Ayurveda and everything. Like, many, even my grandmother used to say some. She had some benign growth in a ear, and
Ah,
her grandfather's father used to take a
yeah,
plant leaves and used to and
yeah.
it just came
I
off.
understand. Yeah, yeah, I understand.
So
Lot of scope, but DM, if you want, you can do. Otherwise, you can do funded research. Ayush is giving lot of funding to do herbal drug, tie up with Ayurvedic physician, get funding, and do
okay, ma'am. So this
proper
was my
research.
doubt. Like, is it necessary to do DM clinical pharmacology for drug development, or is
No,
there any other
not
alternative way?
no. DM is not compulsory, but definitely it's a value add. I have interacted with DM people; they are really good, and they have they develop in a different way. But it's not mandatory, I feel. Rather than that, I would encourage you to apply for funding and take up academic research and validate Ayurvedic products in proper disease condition, because ICMR is promoting holistic treatment.
Yes.
CBME and what is it? NMC is also encouraging holistic. Okay. So I'll take up questions from others, Dr. Revati, if you
Okay.
permit.
Thank you so much, ma'am.
Thank you. Anybody else? Yes. Irana has asked, yes, trapezoid rule. I will explain that in the next session. Good question. For trapezoid rule is the trapezoid formula for calculating area under the curve in pharmacokinetics. Maybe next session, definitely we will take it up. Yeah, somebody said the target disease causing target like COX in inflammation. And okay, anybody? Yes, Amit, you can ask. What is your doubt? Please go
Yes,
ahead.
ma'am. So I want to clarify regarding the field visits and industry visits you mentioned.
Yeah, yeah.
Now, I am doing my MD Pharma at Bangalore.
Hmm.
So, though I have a house in Bangalore, so generally, how will these field visits be if for somebody who is not based in Bangalore, and
Not
how would they
stays in,
get?
yeah, yeah. Last time we had 2 candidates who came from Salem and Chennai. They came the previous day. You have to make your own arrangement to visit. You should come directly to the industry. You have to make your own arrangement to stay, and you can. We will tell you where it is. We join there, and then we continue. Morning one industry we went, then we took a lunch break. Then afternoon we visited another industry. Then next week we came to another industry, like that. So you will have to come on your own.
Yeah, no, not regarding the traveler stay. More
Uh,
of would it be like generally on weekends when we don't have, a
no,
college and all that, or would
no,
it
no,
be?
it will be on a weekday, because the people who are working there, it depends on them. They don't allow us to, I mean, they are not free on weekends. So we
I
have to go on a weekday. You have to take permission from your department.
Okay, sure.
Yeah. Okay. Any other questions? Any other questions? So I have put the post test and one more question, Dr. Ramneni. I will just, how you think now we discussed about all this? Dr. Ramneni, you are there?
Yes, ma'am.
Yeah, you are there, no? Yeah. So how do you think the students should prepare themselves to equip, what do you think? You are a senior, you're taking exams in August, right? Now.
Yes. It's okay.
Yeah. So how did you gear up with all your commitments, and then still you made time to make notes and all that? How did you do that? Is it possible for all students to work, put extra efforts there? And thanks, Amit.
Yes, ma'am. I think focusing on the academics also is very important. It will
Important.
help build skills like preparing for the seminar, how to prepare a slide,
Okay.
then trying different ways how to improve that slide. And
Okay.
thesis also, how to improve the thesis, how to improve the protocol,
Yes.
finding different ways.
Okay.
And learn statistics on your own also. Do the thesis statistics on your own. And
you
then,
suggest let them do it on their own. Okay, that's a
yes,
good
yes. And then, of course, take help from the statistician, but first try with yourself.
Try. Okay.
And then I did myself. So, and of course, after validation from the statistician, I submitted. But it should, once we try ourselves, it will be a learning experience.
It will be.
And
Yes.
similarly, publishing and presenting and writing, everything can be learned.
Okay.
And then also, you can compile the pharmacovigilance cases and probably submit them as a case report or case series.
Yes.
Yeah, that will also improve the presentation skills and reporting skills. So that is and of course, engage in the various conferences and with other candidates of other institutes.
Yes.
Interact with faculty of other colleges also, like that. So that is my.
Great. Yes. So I think you have got the gist. Taking everything very seriously. Do not relax. Be on the job. Take every activity in the department very seriously. Taking lecture, giving seminars, journal clubs. Get completely involved in that. Do not take pharmacology lightly, isn't it? Because I had one student who said, when I asked her to collect, I don't mind, now she's a good friend of mine and we share very good relationship. That time she was very angry with me, and as HOD I said, go and collect ADR forms. Ma'am, I have come to pharmacology to relax. I did not want to do duties, and therefore I came to pharmacology just to relax. You don't make me run around and collect ADR reports. But that helped her to get a good position in a big company, and now she's in a big position in pharmacovigilance. So it matters. So whatever teachers tell, and every seminar topic, these are additional topics, but going through the content, like you're talking about antihypertensive drug, you're talking about a new recent advances in something. Everything is important. You will realize the value when you come. It's not only these added hi-fi things or glamorous things that make meaning. Everything matters, so take it very seriously. So I think if you have done the post test, we wind up here. Priyadarshini, you had a doubt? Priyadarshini, you had raised your hand. Yes, Priyadarshini. Yeah.
Ma'am, can you brief the outline of the 6 months course, ma'am?
Outline. Yeah. Actually, I wanted to share. Okay, so how it will be is every Thursday we have 6 to 8. Today it was introductory entire session. Me and Harshad, Dr. Harshad, we thought introductory, but still it was content was there. Next session we have 2. So every week we will have 2 speakers. Okay. And at the end of the session, today's will go on the YouTube. Rest will be PPT will be shared. Every session we share the PPT, and we will have pre-test and post-test, and every Thursday only. but we want you to be there online on video also, because the speakers would expect that, would like to interact. That is one. And then in between